So now that we have started replication, we can talk about elongating. So our elongation is the second step. So we have initiation, which is our start at our origin, then we have elongation, and then termination. So during elongation in DNA replication, we have our addition of nucleotides, and it occurs, as we mentioned, at a rate of about 1,000 nucleotides per second. Remember, DNA polymerase III can only extend in the 5' to 3' direction, which means it can only add a new nucleotide to the 3' end, the 3' hydroxyl group at the DNA or at the replication fork. So our DNA double helix is anti-parallel, which means that one goes 5' to 3', the other one 3' to 5'. So we have this issue, as we mentioned, where we're not able to add on to a 3' end on the lagging strand. This is where that's mentioned in words. So this continuously synthesized strand is known as the leading strand. So our leading strand, as we saw, we can just start at the beginning, which actually has a primer also. We have a primer there, and then DNA polymerase III can go in and can just continuously add as our replication fork continuously moves to the left in our image here, and it moves to the right as well. So then we have our leading strand, which was our top strand that we saw in the image, and as we were going through, we have our leading strand is continuously adding. So then we have our other strand, which is complementary to the 5' to 3' DNA, and that grows away from the replication fork. And so as it's growing away from the replication fork, we have our DNA polymerase III has to keep moving back. So it can replicate some, and then it has to go back to the replication fork where it's separate, and then it has to complement some more, it has to replicate some more, add nucleotides some more, up until it catches up to its primer that it already started at, and then it has to go back to the replication fork again and then do some more. So it has to move back toward the replication fork to begin adding bases to a new primer in the direction away from the replication fork. And then it bumps into that previously synthesized strand, and then it has to move back again. So these steps produce small DNA sequence fragments known as Okazaki fragments. And they're each separated by this RNA primer. And so the strand that has these Okazaki fragments is called the lagging strand, and its synthesis is discontinuous. So we have continuous on the leading strand, and we have discontinuous on the lagging strand. So let's actually look at that image again, and then we can talk about the rest of it. So again, we have our leading strand up here, where we actually start with a primer here, and then we add to the 3' end, and our DNA polymerase is able to just keep going. And then remember, our replication fork keeps getting further and further to the left. So then we're going to go in, and then all of this, this guy right here, this DNA polymerase on the leading strand, is just going to be able to keep going and going and going as our topoisomerase II goes to the left and opens and unwinds the next part, as our helicase moves to the left and pulls apart, unzips the next piece, and as our single-stranded binding proteins move to the left and hold it open, this DNA polymerase III on the leading strand is able to then just keep going as the replication fork keeps moving to the left. On our lagging strand, however, we have these Okazaki fragments. And so if we can imagine for a moment, I'm going to try to draw on this, if our fragment, actually we'll start down here. So if our replication fork was here to begin with, then we would have had our primer attached here, and then we would have had DNA polymerase III adding nucleotides as we go along this way in the 3' to the 3' end. And then we would have this primer here. And then our replication fork, as it does, is going to be moving to the left. So then it moves to the left. And then, since our DNA polymerase III is going this direction on this lagging strand, now it has to back up, because now it would be here, right? Here's the primer, and then this would be right here. So it's going to have to back up and put a primer here, and then start adding nucleotides this way. So then, let's see if I can cancel that. Great. So then we keep moving to the left. So then we would have our next one that would be here, and we would be adding the primer here, and then keep adding. So then what we end up with, as you can see here, are different fragments. So we would have a section that has a primer, and then nucleotides, and then another section that has a primer, and then nucleotides, and then a primer, and then nucleotides, as our unzipping keeps going toward the left. So we have those chunks are called Okazaki fragments. And then we have our DNA polymerase 3 that's continuously doing that, and then we keep adding our RNA, our primers, RNA primers, and we keep going through that process. So let's talk about what happens to those Okazaki fragments then. So the overall direction of our strand, we have, it mentions here about this sliding clamp. So this is a sliding clamp that's going to hold the DNA polymerase in place as it continues to add the nucleotide. So it's holding DNA polymerase. Maybe we'll see a video where it's kind of this ring structure, and then DNA polymerase is within here. And then we have our strand here that DNA polymerase is, it's kind of overlapping, but is adding the different nucleotides here. This would be our leading strand because it's going this way. But it's adding our different nucleotides in this direction. But our DNA polymerase is just creating that bond between our OH and then our phosphate. So it's just creating this bond here that's going between the two nucleotides. So it needs to be held in place as the nucleotide is being brought to the position. Then DNA polymerase 3 is going to go pchink and make a bond to it, and then it's held by this sliding clamp to slide it down this way again, and then another nucleotide comes along, puts it here, and it goes ka-chink and makes that bond, and then it keeps moving down. So we have our sliding clamp, ring-shaped protein that binds to the DNA and holds that polymerase in place. So then we also have, as we're moving to the left, remember, I can pretend that this is a replication fork here. We have topoisomerase 2, which is kind of unwinding. So we have this supercoiled DNA, and then it's kind of straightening that out for us. Not only does it hold it open and kind of stretch it out so that it's not so coiled up, it also prevents the overwinding of the DNA double helix ahead of the replication fork. So as we're going through and we're opening up this section here, if you've ever played with, again, those telephone cords from a really long time ago or something like that, there are bracelets made out of that and things like that. But if you unwind one section, it supercoils or makes other sections overwinding. So it makes other sections tighter. So then what topoisomerase 2 does also is it makes these temporary nicks in the DNA. So it'll go up here, and then it'll make a cut in the DNA, and then it will unwind all of this that is wound up, that's overwound, and then unwind here, and then it'll attach it again so that it's not so wound up. And then it continues on to keep making things straighter or more opened up for our helicase to come in and unzip, and then all of the other proteins and enzymes to come in. So as our synthesis proceeds, the RNA primers are then going to be replaced by DNA. So back to the Okazaki fragments, we have all of these little chunks of RNA primers then with DNA. So we need to get the RNA out of there so that we can fill it in with DNA. So the primers are removed by exonucleases. So our exonuclease activity of our DNA polymerase 1. So remember, we have DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. DNA polymerase 3 is this guy that's making those interactions or making those bonds between the hydroxyl and the phosphate. And DNA polymerase 1 is going to go to our Okazaki fragments, and it's going to take care of these primers. So it's going to go in, it's going to cut out our RNA primer, get rid of it, and add back in DNA nucleotides. So it fills in those gaps. However, there are still nicks that remain when this happens. Because again, it's a polymerase, it's a DNA polymerase. It can only add to the 3' end. So if I pretend that this is like a zoom in here, we've got our 3' end here, and then let's say that's a nucleotide, our 3' end here, and then let's say this section here is where we've taken the RNA primer out. So our DNA polymerase 1 can go in and it can add nucleotides to here, but then we still have this broken piece here. It can't add another nucleotide, but our DNA polymerase 1 cannot make that happen. It can't make a bond there. So we have an enzyme that goes in and does that for us, and that's the DNA ligase. So our DNA ligase goes in and it makes the bond between this section that's been filled in and then the next section that already exists. So it goes in and it makes that same type of bond, or covalent phosphodiester link, between this 3' end here and then the 5' end on this end. So let's take a look at that in our image, because they have shown that here. So we have our first Okazaki fragment because this would be where the replication fork started. Then our second one, because as our replication fork keeps moving to the left in this image, then we have our Okazaki fragments and it keeps backing up. So they're showing here, there's a full RNA primer here, and our Okazaki fragment is this DNA chunk here. And then you can see that they have DNA polymerase 1 going in here, and what it's done is it's taking out, see how small this is? It's taking out an RNA nucleotide, and it's replacing it with a DNA nucleotide as it goes along in this direction to the right. So then what they're showing between Okazaki fragment 1 and Okazaki fragment 2 is that now DNA ligase has to come in and fix this one bond that hasn't been made in order to make a continuous strand there. And so what it's showing in the blow up here is that our DNA ligase is going to come in and it's going to take this hydroxyl group and this phosphate group and make that phosphodiester bond in order to now then have our continuous strand of DNA on that end. And then this constantly is happening. As our replication is continuing, our fork keeps moving to the left. We keep making more and more Okazaki fragments on our lagging strand. And then we keep going back in with our DNA polymerase 1 and then our DNA ligase in order to fill in where our DNA primers are, or our RNA primers rather, and then our DNA ligase is going to fix that last little nick, that last little piece that needs to be taken care of. So then we run into, or we end up with, our third piece to our DNA replication. So the first is initiation, then we have our elongation, and now we have termination. So once we have replicated our whole chromosome, we're talking about bacteria in this case, even though I keep saying we have our termination of DNA replication. So we know a lot about the other steps. We don't know so much about the termination process. What we do know is that our DNA of our prokaryotes are circular. Since they are circular, they end up being concatenated, which means that the circular DNA chromosomes are interlocked and they have to be separated from each other. So remember, in eukaryotes, we have long strands of DNA. So there are single strands here. Let's say they're not single strands, but meaning they're straight strands. They're not circular, right? So our bacterial DNA, for example, prokaryotic DNA is circular. If you took our DNA, it would be long strands, long straight strands like this. So when we go in our own DNA, for example, we break these bonds and we end up with our DNA like this, we don't have any problem with things being connected to each other. There's no intertwining, there's no interlocking. But once we go in here, then we have a process where we're adding DNA strands in here, and things can get a little mixed up here. So then what we have is our topoisomerase 4, which is going to go in and break some of these strands in these DNA molecules. It'll go in, do some nicks, and then it'll separate the strands out and then seal them back up. So then they're not going to be attached to each other, so that we can have one strand over here and one strand over here, or one chromosome, not strand, it's double-stranded, but one chromosome over here, one chromosome over here, so that we can then go through binary fission and make sure that they get into separate cells. So the resolution of concatamers, those are the enzymes that will go through and do that, is an issue that's unique to prokaryotic DNA. So remember that that's because that's a circular chromosome rather than eukaryotic DNA, which are straight chromosomes. Not when they're actually in the chromosome, or not when they're actually in there. They're all wound up around histones and compacted and things like that. But as they're stretched out, we don't have this issue of them being an actual circle. They can be stretched out into long long straight is the word I'm looking for, long straight strands. So what we see is this problem in bacteria rather than in eukaryotic organisms, so prokaryotes. Here we have a nice little chart for all of the different enzymes that you should be familiar with and what they do. So the enzyme or the factor, and then what they do, and we've talked about each one of these. Do make sure that you understand the difference between DNA polymerase 1, DNA polymerase 3, as well as topoisomerase 2 and topoisomerase 4. And of course all the other ones you should know as well. It's just those can maybe get a little bit confusing because they're the same name, just a different number.